happy to see you all, and welcome to OmicsLogic. So what we'll do today is we'll kind of briefly talk about this upcoming program and explain how you can use various resources that we've prepared for you to learn about biomedical data analysis, machine learning, as well as some coding in R and Python. So first of all, why machine learning? Why data science? So as you probably know, some of the highest paying jobs and interesting tasks are being prepared to tackle data in a variety of ways. And especially this is happening in the world of biomedical discovery, pharmaceutical, biotechnology companies. And the major objective for these companies is to take biological insights and translate them into applications. And a lot of times, to look deeper, just like we look in the microscope to understand the details of a biological sample, today we turn to computational analysis because the data is becoming more and more complex, large, variable, and essentially knowing how to use some of these tools can be a major component of your job description, or it could be something that just drives your career and allows you to explore data sets and gain meaningful biological as well as clinical insight. So we'll talk a little bit more about the types of applications in a little bit. So what we want to do in this program is really focus on some of the applications of data science as well as machine learning to different types of data. These data sets could describe challenges in diagnostics. When we talk about clinical applications, they could be discovery of targets that could be applicable to biotechnology or pharmaceutical companies. It could also be for basic research where we look at environmental samples, clinical samples, animal models of human disease, and try to understand what is the data telling us about a particular condition, phenotype, or biological process. Now, I think the most exciting thing is that there are huge amounts of data available for anyone to learn from and analyze on repositories like the National Center for Biotechnology Information, or NCBI, where you have layers of information from processed information that has already been analyzed and interpreted and put into review articles or publications, but including all the way down to the raw data. And especially for the raw data, recently there has been an analysis, and in 2021, we're over 15 petabytes of SRA data, which is raw data, raw sequencing reads that have been deposited on NCBI. So what is 15 petabytes? 15 petabytes is 15,000 terabytes of raw data, raw sequence archives. Now that includes some process data as well. There's over 100,000 public data sets for GEO, which is gene expression omnibus transcriptomic data. And there are a lot of over 6 million GenBank sequences, which means over 6 million individual species that have been sequenced and their genomes have been assembled. Now, a lot of this is driven by what's happening right now. What are the hot topics in research, including clinical research, including basic research? And one of the major drivers, I think, right now is the pandemic, which has brought about almost real-time sequencing to the forefront, really allowing many of us to not only learn about the impacts of the pandemic, but also look at how we can contribute to the understanding of this pandemic. Now, when it comes to such a diverse series of data, we need to understand what are the properties and challenges associated with each one. Omics data is a variety of comprehensive views of a particular type of data. Now, a lot of times this is generated using what's called high-throughput technologies. These technologies include phenomics, genomics, epigenomics, and transcriptomics. And all of these are relying on high-throughput or automated technologies to generate huge collections of data sets. Now, because they are so variable and they speak about different molecular properties, you have to rely on computational technologies to be able to learn from these data sets, analyze these data sets, explore these data sets, and make use of these data sets. And these computational technologies are basically rooted in data science. And so in this program that we're going to introduce you today to, we're going to focus on some of these data science skills that you need to have to be able to efficiently process, analyze, and visualize such complex biomedical data. So what are some of the things that we're going to cover in this program? First of all, we're going to talk about analytical methods, how to visualize and analyze the data complexity and reduce it so that we can extract meaningful insights. We'll introduce you to the terminology for machine learning and artificial intelligence and biomedical discovery so that when you read the paper and you hear about a method, you know what it does and you know what it means. We'll also provide you with some project examples so you'll see how other people have used machine learning. Now, machine learning is used in many different ways. And so you'll see how it's not just clustering or classification. You'll see how different types of machine learning methods are actually used in a variety of tasks related to the whole process, from getting raw data to then making it useful for analysis or structuring it, cleaning it, preparing it for analysis, and then finally turning it into a model or learning from it, essentially using the complex patterns in that data. Now, as a result, what we expect the participant to gain out of this program is to really have an understanding of how machine learning algorithms are used in analysis of omics data. You'll understand the differences between different types of machine learning models and different types of problems that they can be applied to. You'll also see practical examples. So you'll have a sense of how other experts have used this and what are the specific challenges that they had to address to make their project work. And we will introduce you to some of the popular and user-friendly Python and R packages so that you gain practical skills. You can actually use these skills to code yourself, and you will be able to read other people's code, understanding what packages are being used and how to use them yourself. And so these are going to include some examples that we'll actually do today in this session so that you get a sense of how can you get all of this done in a short period of time. So the data science program, the OmicsLogic data science program, is really structured around these four different things: understanding the various omics data types, learning to explore them, then to try big data analysis in the context of a project, and then to understand the link between data and biology. How do you actually master biological interpretation leveraging some of these tools? So let's talk a little bit about the syllabus. So the syllabus of this program is organized into three different parts: introduction, methods, an in-depth review of methods, and finally, the project section. So altogether, we'll have three different sections. The first one is we'll introduce you to the relevant terminology and key concepts to refresh your memory and get you started so that you're well oriented in this whole field of omics and machine learning. The second one, we'll talk about methods that will give you a practical sense of commonly used methods and functions for analysis of omics data, starting from exploration to statistical testing to supervised and unsupervised machine learning, as well as deep learning. And then finally, some project examples so that you have a sense of applications for such methods. And after you complete the full program, we hope that you will have the confidence to understand and analyze data that you can conduct independently to a data set of your choice. And we'll also introduce you, by the way, to some examples at the end so you can kind of try it out before you do your own independent work. So the program itself is going to combine interactive sessions, just like we're having today. In each session, you will find additional links to asynchronous resources that review and enrich the covered topics. So it's the live sessions plus the self-guided study materials that you can practice and go deeper into if you want to learn more about the technical, the theoretical, or the application of the particular topic. And as we kind of talk about a lot of topics in general, I want to kind of give you a high-level overview of each one of these topics. So a lot of what we are talking about when we say words like transcriptomics, genomics, or metagenomics might be confusing to some of you. And so we will start in the beginning to kind of explain what is the link between omics data, the biological features, the molecular features that they describe, and how can the data that comes from an automated process like sequencing can be used to essentially prepare it for analysis. So we want to link together the biological, the technical, and the data components together to kind of make sense of the important terminology and give you a practical sense of what does this data look like and how does it work in terms of using it for data analysis. Then in the second part, we'll talk about the machine learning topics, unsupervised and supervised learning. How are these methods used for various tasks such as visualization, organization of data, finding signal in noisy omics data, as well as things like predictive analysis and feature selection. So to use these methods, you will need to understand how they work. What are the inputs? What are the variables? What are the different settings and available packages that combine multiple functions that make the methods that we describe easy to deploy, right? So you can understand the method in detail, but then as you want to deploy it, you want some user-friendly or some kind of a shortcut to essentially launch and train that model. So that's what you will learn in this second part. You will learn to load data, select a method, and run it to get an output that you can interpret for that particular model. Using these principles, we will then review additional methods that are useful for biomedical discovery that include network analysis, such as pathway analysis for biologists, generalized linear models that are used to distinguish between expression profiles of transcripts, for example, and complex transcriptomic data, as well as deep learning models that can be used for specific types of situations to improve accuracy and decrease feature engineering, which is a lot of what conventional machine learning relies on. And then we'll also talk about the need to combine various machine learning models into a series of steps to achieve more complex tasks. For example, when we talk about single cell data. And finally, we will talk about specific use cases that can show you how the methods we reviewed have been used by researchers to find meaningful insights in complex experiments, biological samples, animal models, or patient data. And so with the few examples we review as case studies, you can also then develop a project of your own or follow our asynchronous tutorials to reproduce an analysis that you can improve upon, turning it into a novel study. So what are some of these projects? We'll talk about cancer macrophages, which is an important part of the immune system and the immune response to tumor growth. We'll talk about different types of pandemics like enterovirus, malaria, or Ebola virus. We'll talk about modeling cancer precision medicine, where we can find biomarkers and learn how different patients respond to different treatments. We'll talk about tumor microenvironment, but we'll also introduce projects that are not clinical in essence, things like drought resistance or different stresses that are important for crop production and environmental preservation. So as a result, I think that you will really get a full overview of the data science kind of a process in biomedical research to understand how different stages of analysis work together to help define the hypothesis, as well as prepare the data for meaningful application of machine learning for big data practical challenges. So as a result, you'll be able to take a data set, make it useful, and then explore it from a biological perspective to understand really what does it mean. As a result of completing all of these different steps, you will also get a certificate. So as already mentioned by Sonalika, you will get the certificate after completing all of these different steps on the OmicsLearn portal. So now to kind of explain how all of these different resources are linked together and how to use them, I want to give you a practical sense of how this works. So for those of you that have not signed up yet, please go on to learn.omicslogic.com and sign up and create an account. And then I will show you a couple of just really simple practical examples to illustrate what we will do. So this is the page where you have registered on the Learn portal for this webinar. And as you can see, it explains everything that I've described today. And right here on the top, if you already have an account, we can go to the courses page. And what I will do is I will take one example from the asynchronous materials that we will go through to just kind of illustrate what will be going on. So if you go into the search tab right here on the course page, if you go into the search, go to data science. And you will see here BioML in Python and BioML in R, as well as some additional resources that you can look at. But right now, we'll just take one example from this introduction to data science in R. And let's take a look at some of the basic tasks that you have to get started with as you are considering this program. So this is the course. I'm just going to paste the link again in chat for those of you that want to follow along. And in this course, this is again R coding course 2, Introduction to Data Science or BioML. You will see all of the different courses that we have. There's about 10 courses here that go from very, very basic things, or how to load data and check for variable data, all the way to advanced supervised machine learning, right? So let's take a look. I'm going to use this course right here, or this lesson right here. Anyone without a paid subscription can access this lesson. It's called Data Processing and Visualization. And basically what it talks about is once you have a table, of gene expression, a table of OTU taxonomy in metagenomic sequencing of the microbiome, or whether you have some kind of frequency of mutations across the genome of a virus, you will have a table. In this table, you will have different types of data. This data can include continuous numeric data, such as transcriptomic gene expression. It could have names of samples and names of genes, or OTUs. And it can also have categories. So these could be groups of samples that maybe define a particular type of samples that you have. Now, to analyze such data, you can explore different methods of visualization that summarize the data in a meaningful way. And that can include crosstabs or pivot tables. They can include box plots that summarize statistical properties of each sample or each group. And they can include continuous data like scatter plots that allow us to find relationships and organization of data that could be potentially interesting from a signal standpoint. So as we go through this introduction, you'll kind of learn about some of these methods, and then we can jump into the breakdown of this code. Now, what's important is, first of all, loading the data. And so here you have an example of how you load a data set that is already on GitHub. Well, this particular example is on our portal, but we'll also be introducing you to how to use GitHub to kind of manage and organize your code as well as data sets. Then you can pre-process and transform your data for analysis. So here you can see that we can, for example, prepare the data matrix from the data frame that we have to create some numeric data for analysis. Then you can also load a library. A library is just a compilation of different functions that are going to be useful for the analysis and streamline your analysis. And in that data, you now have an option to create a visualization, like a box plot, right? So after we learn about how this code is organized and what are the different elements of this code, we can actually go in to practice it yourself right here in the browser. So let's go into practice it yourself, and let's try over here to run this analysis. So you see that now you have a console, you click on run, and after this step is complete, you have a message success running code, and essentially you get an output that was predefined for you to make sure that you're following the steps. Now here, nothing really has to change. It's already made for you. What you can do though, is you can check what's in this data. So let's go in here and we'll edit it, and we will put in head df, which is the name of our data frame, to understand what is in our data. So after we have calculated the mean, which is right here, we now see what is in our data. Now, this is a little bit difficult to understand. As you can see, there's a lot of different columns. There's a lot of different numbers. It's not very clear what do they actually mean. A more useful way to look at this data is to actually visualize it. What's in it? And so here we have a scatter plot, box plot, histogram, and heat map, but all of it is connected into one single piece of code. And as a result, we might be a little bit confused as to what exactly are we looking at. So let's modify this a little bit. Here we also have descriptive statistics converted into matrix, draw a box plot, a heat map, and a histogram. So let's start with the box plot. So we'll just remove this, run it, and now we have a box plot. Now, what is a box plot? A box plot essentially shows us how the data for each sample that we have on the x-axis can be summarized using the interquartile range, maximum and minimum values, outliers, as well as the median line for each one. This gives us a sense of what's in the data that is fairly complex. We have here a bunch of samples, and inside each sample, we have certain level of variability that we should be using to understand differences between what we see right here, which is the names of groups or conditions that we're trying to interpret. Now, what's important about this visualization is that it should immediately give us a sense of how ready is this data for downstream analysis, right? So if we want to analyze a difference between normal-like samples and, let's say, basal or luminal breast cancer subtypes, we want to understand that within each group, there's some consistency that we can rely on to make sure that these samples accurately represent the condition that we're studying. And that is really what this whole course is about. This course is going to go into the detail of the code, but also link it with the biological question that you're trying to ask to really understand what is the process of going from raw data to an answer to a question that is either biological in nature, clinical in nature, or has some interpretable insights for somebody working in industry like biotechnology or pharmaceutical. So with this, I want to really make sure that it's clear that in the context of the program, we will cover a lot of topics in sessions like we did today, going through code and running examples. At the end, you will be able to download all of this code. You can copy it, and we will use what's called Google Colab to make sure that all of the code that you work on is not only tested automatically on our platform, but also is reviewed by someone so that at the end you come out with a whole series of full code from beginning, loading, exploration, finally to training a machine learning model, and then interpreting your results. And all of that, by the way, is going to be integrated on your own profile. So at the end, when you share your outcome, you will see all of the different projects and all the different courses that you have completed. You will also have the option to develop an independent project. So here, after you have a certain number of points, after you've completed the minimum requirements, you will be able to apply this to a data set of your own, essentially turning it into a mini publication that starts from submitting this project right here on this Learn portal that will guide you through all the different steps that you need to take. And then you can actually use this to maybe go for a poster presentation or a full publication if you have a meaningful project. So with that, I want to make sure that I answer any questions that you might have.